Welcome back for another deep dive. Today, we are going to be tackling Arcanobacterium Haemolyticum.
Oh, interesting.
Yeah, it should be pretty interesting. So this one, I actually pulled some research papers for this one, so this could be a little bit more in-depth than usual.
Cool.
And we've also got some notes from Notebook LM and some insights from MicroWiki and Microbe Kansas to kind of round things out.
Excellent.
So get ready to see this bacterium in a whole new light.
It's a great choice for a deep dive. Arcanobacterium haemolyticum is really a perfect example of how complex the microbial world is.
Right.
I mean, we're talking about a bacterium that can just be hanging out in our normal microbiome. And then suddenly it can become pathogenic.
Yeah, that's kind of a theme we've been seeing lately in some of these.
It is.
Pretty wild. So let's start with some basics.
Sure.
Can you remind us what makes Arcanobacterium haemolyticum unique from like a microbiological standpoint?
Yeah, so it's a gram-positive, facultative, anaerobic bacillus.
Right.
And something that makes it really stand out is its shape.
Okay.
It has this really distinctive branching pattern that you just don't see in a lot of other bacteria. And of course, like you said, it has these two subtypes, rough and smooth.
Right.
And you can tell the difference just based on how their colonies look, which is pretty cool.
Oh, so they actually look different.
Yeah.
That's so interesting. So it's a bit of a shapeshifter then, huh?
Yeah, you could say that.
I'm curious, does that actually affect how it behaves in the body? Like, does it being rough or smooth matter?
That's a great question. And actually, it's something that researchers are still trying to figure out.
Okay.
But it is interesting because we see the smooth subtype more often in wound infections. And then the rough subtype seems to prefer the respiratory tract. And there's a hypothesis that the rough surface structure might help it to stick to respiratory cells more effectively.
Oh, so it's got like little Velcro.
Yeah, basically.
Oh, that's interesting. Okay, so that's morphology.
Right.
And now let's talk about how it actually causes problems, because we know it can cause infections.
Yeah.
But what's actually going on there?
Yeah, this is where it gets really interesting.
Okay.
So Arcanobacterium haemolyticum has these two main virulence factors, arcanolysin and phospholipase D.
Okay.
And they kind of work together in this really cool way.
Cool. So break it down for me.
Okay, so first you have arcanolysin, which is a cholesterol-dependent cytolescin.
Right.
And basically it acts like this targeted weapon. It binds to cholesterol in the host cell's membrane.
Okay.
And once it's bound, it forms these pores.
Ooh.
And these pores really disrupt the cell's integrity. And of course, that eventually leads to cell death.
Oh, wow. So it's taking advantage of the very building blocks of our cells.
Exactly.
Wow, that's pretty sneaky.
It is.
So what about phospholipase D? What does that do?
Okay, so phospholipase D is kind of like a scavenger. It helps the bacteria to get cholesterol from the surrounding environment. And then that cholesterol fuels the arcanolysin so it can keep doing damage.
Oh, so they work as a team.
Yeah, like a tag team.
Oh, that's amazing. It's a powerful combination.
It really is.
Now, this cholesterol dependence, is that common? Do other bacterial toxins do that?
It's actually pretty rare.
Really?
Most bacterial toxins don't rely on cholesterol in this way.
Wow.
Which is why Arcanobacterium haemolyticum is so interesting.
Okay.
I mean, it makes you wonder about how it evolved and stuff like that. And it could even help us to develop more targeted therapies.
Yeah.
Because it's so unique.
That's super fascinating. So we have this bacterium with this pretty unique weapon.
Yeah.
But what does that actually look like for people? Like, what kind of infections does it cause?
Well, the most common one is pharyngitis.
Okay.
Sore throat.
Okay.
Usually with fever, swollen lymph nodes, and a rash that kind of spreads out. But it can be tricky because the symptoms are super similar to strep throat.
Oh, so it could be misdiagnosed.
Yeah, definitely.
Okay, so is there anything that clinicians should be looking out for? Like any telltale signs?
Yeah, so the rash is a big one.
Right.
It usually starts on the arms and legs and then moves toward the center of the body, which is a little different from other bacterial infections.
So it's like the opposite direction.
Yeah, you could say that.
Okay, good to know. So pharyngitis is the most common.
Right.
But what else can it cause?
It can also cause wound infections.
Oh, okay.
Things like skin ulcers, abscesses cellulitis.
Yeah?
And in some rarer cases, it can even cause more serious complications.
Okay.
Like pneumonia, brain abscesses.
Oh, no.
Endocarditis.
Oh, geez. So it can really range from mild to severe?
Exactly.
Which brings up a question, how do you even know if it's causing the infection?
Right.
Because it can just be hanging out normally.
Exactly.
So how do you tell?
That's a great question, and it's a really important one.
Yeah.
Just because you find Arcanobacterium haemolyticum in a sample doesn't mean it's the bad guy.
Okay.
So clinicians really have to look at the whole picture, like the patient's symptoms, their medical history, all of that.
Yeah.
And sometimes they might even need to do more tests.
To be sure.
Yeah, so medical microbiologists really have to be detectives sometimes.
Oh, yeah, it's a puzzle.
Totally.
Okay, so let's dig into some more general info about this bacteria.
Okay,
So MicrobeWiki says that it mostly infects humans.
that's right.
Like, it's not really found in animals very often.
Yeah, it has been found in animals, but it's pretty rare.
Okay,
Humans are definitely the main reservoir.
interesting. So it really likes us.
It seems that way.
And what about where it likes to live on us? Like, what's its favorite spot?
Well, we didn't know for sure,
Okay,
but it tends to hang out in the throat and on the skin.
so it's pretty picky about its host, and it's picky about where it lives on that host?
Yeah, pretty much.
And what about in the lab? Like, if you're trying to grow it, what does it need?
Well, it's a facultative anaerobe, remember?
Right, yeah.
So it can grow in different oxygen conditions.
Okay.
But it really prefers an environment with a lot of CO2, ideally around 5 to 10%.
So it's a CO2 fan.
You can say that.
Before we move on to the exam questions, let's do a quick recap.
Okay, sounds good.
What are the key things to remember about Arcanobacterium haemolyticum?
Okay, so we're talking about a gram-positive
Okay.
branching bacillus.
Yeah.
That's a facultative anaerobe and loves CO2.
Uh-huh.
And it's also beta hemolytic.
Right, so it can break down red blood cells.
Exactly.
All right. Ready to put our listeners to the test with some exam questions?
Yeah, let's do it. Let's see if they've been paying attention.
Okay, so question number 1. A patient comes in with pharyngitis. They have a fever and a spreading rash that started on their arms and legs. What bacteria that is often found in the normal human microbiome could be causing this?
All right, think carefully, everybody. We'll discuss the answer after a short break. All right, so let's think back to that patient.
Okay.
Pharyngitis, fever, and that spreading rash that starts on the extremities. What do we think?
Yeah, that rash is really a key clue here, isn't it?
It is, and remember, we're looking for a bacteria that's often just hanging out in the body, not necessarily an outsider.
Right, right. So what's the answer?
The answer is Arcanobacterium haemolyticum.
Make sense?
Yeah, remember, it can be part of our normal flora in the throat and on the skin, but when conditions are right, it can turn nasty and cause pharyngitis with those telltale symptoms.
Like a wolf in sheep's clothing.
Exactly. It goes to show you, sometimes the enemy is already within.
Okay, on to question number 2. So A. haemolyticum produces this cholesterol-dependent cytolysine.
Uh-huh.
What is it called, and how does it actually cause damage?
Okay, so the cytolysine is called arcanolysine.
Arcanolysine, okay.
And remember, it targets cholesterol in the host cell membrane,
Right.
binds to it, forms pores, and boom, the cell's integrity is shot.
It's like poking holes in it.
Pretty much. And eventually, that leads to cell death.
It's amazing how it uses something that's supposed to be good for us, cholesterol, and turns it against us.
It's really clever.
All right, question number 3. We talked about the 2 subtypes, rough and smooth.
All right.
How can you tell them apart in the lab?
Okay, so this one's a bit simpler. Medical microbiologists can differentiate them just by looking at their colonies.
Really? Just by looking?
Yeah, it's all about the morphology.
Okay.
Smooth subtypes have, well, smooth edges.
Uh-huh.
And rough subtypes have rough, kind of jagged edges.
So it's pretty straightforward.
Yeah, you can literally see the difference.
Wow, that's so cool. All right, question number 4.
Okay.
This one's about diagnosis. What makes A. haemolyticum infections tricky to diagnose?
This goes back to the fact that it can be part of the normal microbiome.
Right, right.
So finding it in a sample doesn't mean it's automatically causing the infection.
So you have to be careful.
Exactly. Medical microbiologists have to be detectives here. They have to consider everything, like the patient's symptoms, their medical history, maybe even run additional tests.
So it's not always cut and dry.
Not at all. You have to look at the whole picture.
Okay, and last but not least, question number 5. Name 2 virulence factors produced by A. haemolyticum, and what do they do?
All right, let's see if everyone was paying attention. We've got arcanolysine, that cholesterol-loving cytolysine that punches holes in those host cells.
Uh-huh.
And then there's phospholipase D, which helps the bacteria grab that cholesterol to fuel arcanolysine's rampage.
They're quite the pair, aren't they?
A real dynamic duo.
It's amazing how much we've learned about this bacterium in just this deep dive.
It really is. We've covered so much, from its unique shape and those cool virulence factors to the challenges of diagnosis.
And we've seen how it can be both friend and foe, a resident of our microbiome that can turn against us under the right circumstances.
That duality is what makes it so fascinating, wouldn't you say?
Absolutely. What are your final thoughts on A. haemolyticum? Anything that really stood out to you?
Yeah, it really makes you think about that delicate balance between us and our microbial inhabitants.
Right.
And if we can figure out what tips that balance, what makes it go from good to bad.
Yeah.
That could really change how we diagnose and treat these infections.
Totally. Totally. Okay. Well, we've covered a lot today.
We have.
Everything from its weird shape and those awesome virulence factors to how tricky it can be to diagnose.
Yeah, and we've seen how it can be both a friend and an enemy, right? Living peacefully in our microbiome and then is suddenly turning against us.
Exactly, exactly. Any final thoughts for our listeners, especially for those medical microbiologists out there working in the labs?
I think a Haemolyticum really shows us that we can't take any bacteria for granted.
Oh, yeah.
Even the ones that seem ordinary can surprise us. So it's super important to stay up to date on the research.
Right.
And to always be really thorough when you're diagnosing.
Yeah, never assume anything.
Exactly.
So to sum it all up, if you're a medical microbiologist and you come across a Haemolyticum, just remember, it can be a bit of a double agent.
Yeah.
It might seem familiar and harmless, but it can definitely turn into a serious threat.
Right. And never underestimate the power of just paying attention, taking your time, and being curious.
That's great advice. Well, thanks for joining us for this deep dive into a Haemolyticum.
Yeah, thanks, everyone.
We hope you learned something new, and as always, stay curious.